Hello everyone, welcome back to my channel Digital Srini on YouTube. And if you are not a subscriber yet, this would be a great time to hit that little subscribe button down there. And also welcome to video 5B as part of our AI and digital pathology series. In the last video, in video 5A, we went from a raw whole slide image to a set of patch embeddings with spatial coordinates, right? So we processed an entire whole slide image. We subtracted the glass part. We only looked at the tissue part. And from within the tissue, we extracted 256 by 256 tiles or patches, if you want to call them. And we converted that into embeddings using a foundational model. And those embeddings basically means a vector of size, in our case, 1024. So at the end of the tutorial, we ended up with number of patches for that slide. I believe we had like 4,000 some patches and corresponding vectors. We also saved to the disk the total number of patches, like for each patch, the coordinates, X and Y coordinates. So we know where they lie within that tissue. So we can go back and do spatial mapping. Now, instead of one such slide, just imagine working with multiple, a few hundreds of them. And that's exactly what we're going to do here. We have 427 colorectal cancer whole slide images. Well, we don't have the actual images. We have the embeddings, like once they're converted to this 1024 vector. And this data set, it's coming from SurGen, SurGen, and SR386 is the data set. So instead of just exploring what the embeddings look like, we are asking a clinical question here. Does the spatial organization of these tissues around a tumor, does it predict whether a patient survived 5 years or not? And again, why 5 years? Because we have that information for all of these slides. Yeah. Now, again, where this sits in our series is obviously right after video 5A. I definitely recommend watching that because it kind of gives you an idea in terms of how we actually read the whole slide image, how we extract these embeddings, and we save the spatial coordinates along with these embeddings. So that's our starting point in 5B, even though we're not loading exactly the same data set. That's why I say maybe you don't need to pause this and watch that video first. You continue this, but watch that at some point. Okay, now let's start by understanding what tumor microenvironment is, right? So when a pathologist looks at cancer biopsy under the microscope, they're not just looking at the tumor cells, they're looking at the entire ecosystem. The tumor actually sits within a matrix of fiber connective tissue called stroma, and surrounded by immune cells and surrounded by blood vessels and regions of normal tissue. And this ecosystem is what we call the tumor microenvironment. And the tissue surrounding a tumor actively influences how it grows, of course, right? How it invades the surrounding structures and how it responds to treatment. So a tumor that grows in, let's say, compact, dense nests with little stroma behaves very differently from the one that is embedded in extensive fibrous stroma. So what makes this interesting for AI is the scale. So a pathologist can estimate the stromal content of one slide in a few minutes. They're trained to do that. They're highly trained, highly skilled. But obviously the assessment there is qualitative and variable between observers. So we can quantify it consistently across all of our 427 slides information that we are looking at later on using the classifications from the embeddings. And the most studied spatial feature in this context is the tumor stroma ratio, or TSR. Okay, we'll talk a little bit more about this later on. It's the ratio of tumor classified tiles to the stroma classified tiles. How many tiles are tumor classified? How many tiles are stroma classified? And multiple published studies in colorectal cancer show that stroma-rich tumors, low TSR, have worse 5-year survival. So low TSR, worse 5-year survival. So we'll test whether we can recover this signal computationally. We'll test whether we can actually kind of validate that. Yeah. Now, I just mentioned about TSR, just a bit more about it. Okay, it's conceptually very simple. You classify every tissue tile in a slide as tumor or stroma. right? You classify them as either tumor or stroma. And you count them and divide, that's it. A value below 1 means stroma dominates. A value above 1 means tumor dominates. And in our data set, the range actually goes from 0.1, I think, did I put that somewhere? In our case, it goes from 0.1 to about 26 point something. Yeah, a span of like 2 orders of magnitude across the entire cohort. So the grid diagrams show what stroma-rich slide looks like at this tile level, right? Almost entirely this greenish or teal color with just a very few red, like a red rim right there. And the right-hand side, of course, it's exactly the opposite, right? So this is tumor-dominant slide, almost entirely red with a thin stromal boundary. So that's tumor-dominated. So you have a large tumor with like a very thin stroma. Stroma dominated, like tumor, like right there. Now, in addition to TSR, there are certain features that we compute, right? So we compute 4 other spatial features in our case. Spatial entropy. By definition, entropy means it's something like has to do with disorder, right? It measures how uniformly tumor tiles are spread across the slide. So high entropy means high disorder, means high tumor is everywhere. Low entropy means it's concentrated in just one region. And stromal adjacency is another one. So is intended to measure how much stroma is in direct contact with tumor. Yeah. We'll discuss like an honest problem that we run into later on. We'll not discuss the actual results here. Let's jump into code and then discuss the results over there. But I just want to, there is an issue that we'll talk about in terms of extracting some information here. Now, before jumping in, one other thing that I want you to be aware of is the Zarr format. I'm not sure how many of you worked with Zarr format in the past, but we are working with Zarr format because the SurGen embeddings that we download, they actually, this is a 16.8 gigabyte file, by the way, very large file that you need to download to get access to this data. And it's easy to get access to that data set. We'll talk about that in a minute. But each slide within that zip file, it's multiple slides, 400 plus slides, right? Each slide is stored as a separate Zarr array. Think of it as something like NumPy array, but it's a Zarr array with 2 data sets in that Zarr array. It's features 8,249 in this example. You see, that's one Zarr file. And it has features, in this case, 8,249 tiles. And for each tile, you have 1024 embedding, right? And it also includes, for each tile, the X and Y coordinates, the position of that specific tile. So Zarr is a chunked array format, right? So unlike a NumPy array, the last tutorial we stored the NPZ file. And you have to fully decompress that file before accessing. You load, you decompress, you access that. Zarr actually stores each array as compressed chunks. Each of these arrays are compressed chunks. And why is that good in our case? We can read one slide's data without decompressing the other 426. So I don't need to load entire thing in memory. I can actually load one. I can investigate. I can look at that. So that's one of the advantages of Zarr. And even better, Zarr has a ZipStore backend that reads directly from inside a zip file. Meaning I don't even need to uncompress all this zip file and then store all the files into the disk. I just downloaded this file and I'm accessing these files from within that zip without unzipping anything. So this is another advantage. I added like a couple of things here because I had to go through this code and experiment a few times. And I realized, okay, the correct pattern to actually open this is zarr.open_group is the right way with the right path argument. Again, if that's something you can learn yourself by asking ChatGPTs and Claudes, and it took me a little longer than I expected. That's why I just placed it on this slide. No other reason. Okay, now the tissue classifier itself. Again, one thing I want to highlight is what we would like to do with all of this with Zarr. Okay, we have, for example, a given whole slide image. we have like 8,249 features and we have 1024. Let's say you convert that into a UMAP, principal component analysis, and then you have like a UMAP and you're trying to plot them to identify different clusters. We did this in the last tutorial. Now the problem is, in the last tutorial, once we got the clusters, we opened a typical image or a few typical images within each cluster, and then we're like, this is the tumor region, this is the stroma region, this is the background region, right? The problem here is we do not have raw images. We only downloaded the embeddings right there along with some labels, but we don't have raw images. So we have to find a way to kind of label each cluster. I'll talk about that later on once we get to the code, but to compute this, the features, for each tile in each slide, like I was saying, whether it is tumor or whether it is stroma or background, we kind of use an unsupervised approach, like I just mentioned. We kind of do k-means at k equals to 5, and in this case, you see how we see these 5 different clusters. These 4 are pretty clear to me. This one also looks like a separate cluster, but yeah, these 4 clusters. And now the problem is, what is this? What is this cluster? What is that cluster? For example, if we know that this cluster is tumor, this is the background, and this one is like these two combined are stroma or whatever. So once you have this, then we can train a logistic regression. By the way, you can generate this plot not by using all 427 slides, because that would be a lot of data. You can just load a 5 reference slides. That's what we're going to do. We are going to load 5 reference slides with 33,780 tiles right there. And then we run k-means with k equals to 5. And then we assign labels, like we inspect this. And we'll talk about how we are going to assign labels without looking at the tissues. We have to find a workaround. And we'll talk about that once we get there. In fact, let me go ahead and mention the approach. We are going to use clinical correlation as a proxy. Since we don't have images, we are going to use the clinical correlation. We have the clinical correlation information here. We predict the cluster assignment for all the 427 slides. Yeah, once we use clinical correlation as proxy to say, okay, this is our tumor, this is our background or whatever, you train the logistic regression model. And then you classify all 427 slices. That's pretty much, slides. Yeah, the cluster that correlates most positively here with stage and mortality is most likely the tumor, right? So if you have a positive correlation with the cancer stage and also the mortality rate, then you know that, okay, that's probably tumor. That's how we are going to label. That's the workaround that I'm talking about, proxy. In an ideal world, if you have images, then you look at the image, and as an expert, you can say whether that's tumor or not. Now we are going to look at proxy. That's the only caveat I want to give you before jumping in with this approach. Yeah, we are flying a bit blind, and I want to be upfront about that. And also, along the same lines, a cluster with the near zero clinical correlation is most likely the background, right? And what's the other one? The remaining clusters with negative or neutral correlations are stroma. That's my assumption going forward. Okay, now the spatial features that we are going to compute here are the tumor fraction and stroma fraction. These are simple proportions, right? What percentage of tissue tiles fall into each of these classes? And they always sum to 1. So they carry the same rank information as TSR right there. So we compute all these 3 because they have different interpretations in the literature. Yeah, and spatial entropy, already mentioned, it's more nuanced feature. We divide the slide into a 10 by 10 grid. And we compute the entropy, the Shannon entropy value of tumor tile distribution. And a slide with tumor uniformly spread across all grid cells has high entropy. And a slide where tumor is concentrated in one corner, it has low entropy. Okay. Why are we doing that? Because it captures something about invasion pattern that fraction alone misses, right? So if you only look at the fraction, you don't know the distribution, the spatial distribution. And stromal adjacency is intended to measure how much stroma is in direct contact with tumor, specifically the fraction, the fraction of stroma tiles within 2 tile widths of any tumor tile. Yeah. Again, there is a practical issue, like I mentioned earlier, with this feature related to the coordinate scale at 40x magnification. Okay, and I'll discuss about that once we get to the notebook. So, based on the published literature, we expect tumor fraction and the TSR to correlate positively with mortality, and stroma fraction to correlate negatively. Again, based on the literature. Whether our classifier is sensitive enough to recover that signal, we'll see that later on. Okay. Now, in terms of the dataset, we are going to download the SurGen SR386 dataset. Again, this is 427 colorectal cancer whole slide image dataset, and it's scanned at 40x. The previous tutorial 5A, we looked at 20x data set, and that's why we had about 4,000 total tiles. Here you are going to end up with 8,000 or so tiles per whole slide image. This is a higher magnification 40x data set. It also captures the 5-year survival outcome, the clinical stage, stage 1, stage 2, etc., MMR loss status, and mutation data for KRAS and NRAS and BRAF. Yeah. So to follow along this tutorial, you need these 2 files. And the embeddings file, download from Zenodo right here, and the labels file, download from GitHub, the link given down here. And they both cite this. Where did I put this citation? The 2 papers cite are Miles et al. MYLES. And I need my glasses. I thought I put that on the screen. Yeah, right there. Miles et al. Right there. This is the citation in case you wonder. Okay, now I think we are good. Let's not go through these. We are good to jump into our code and have more discussions over there. And the link to this code is again down in description. And this part we have already discussed as part of our presentation. And let's go ahead and get our imports done. And I think everything you should be familiar, especially if you watched the last tutorial. The only thing that's different is probably the Zarr. And of course, Zarr for reading the Zarr archives, right? And also the lifelines. And this is needed if we want to do the KM, fitter right there. We are going to skip this because we'll get there. We don't have the right information that we need to perform this. Again, I'll get to that later on. So all the imports are done. Okay. And all the paths should be done. I mean, this is just defining the path. So we should be all good right now. Now, let's go ahead and load this SR386 dataset. And we load the CSV right here. Again, we are loading this. And let's go down and see. We have 427 cases. We know that. And the survival outcome, 264 were tagged as 0 right there. And that is survived 5 years. And 161 unfortunately died. They did not survive. Yeah. And stage distribution, you have stage 1, 60, stage 2, 153 of them, stage 3, 120, and stage 4, 20. MMR loss, you have 32 cases of MMR loss cases right there, which is basically 7.5% of the entire cohort. And here is some of the Zarr file names that you can find in there. So they are like T001, T003, and so on. These are the different different slides, whole slides. So let's load one of these files and kind of see exactly what's going on. Yeah, it's very important to understand before you jump in. So I am loading one of the files right here, and the files, Zarr files. And by the way, this zip file actually comes with not just SR386, but also another data set. And that's why we are going through this zip file, and extracting everything that has SR386 in its name, and get all the SR386 Zarr root names And once you have that, you know how many you have. And that's what I'm printing down here, SR386. And there are 427 zip files. And we also load the CSV. Remember the second one, the labels, or not labels, the metadata in a way. You can, or labels is probably also right. It's in CSV file, and we have 427 of them, and all of them are matching the names here. Everything's fine. So that's a nice sanity check right there. Okay. And now let's load the embeddings for one slide. That's what I meant to say earlier. Let's look at one slide and understand exactly what's going on before we process all these 427 slides. So here you can see we have by the way, it uses zarr.ZipStore, like I mentioned in the presentation, to open the zip file. Okay, and then zarr.OpenGroup. What's to discuss? Okay, so this is T001. It has 8,249 tiles, and the feature shape obviously is 8249 times 1024, and the coordinate shape is 8249 by 2. And the sample coordinates show values like 76,480, 165,000. These are level 0 pixel coordinates at 40x magnification. Remember, this is the whole slide image. So you have level 0, level 1, level 2, and all that. Level 0 is the native resolution. At that native resolution, what the coordinates are. And that's what these are. Okay. And adjacent tiles have you see the next tile here is 78,494. 76,480. That means there is a stride of 2013, 2000 something pixels, not 256, because the tile size is 256 pixels at 40x, but the coordinate space is at full 40x resolution. I hope that makes sense. Okay, and that stride is one of the reasons why the stromal adjacency metric probably fails later on. Things are not under our extreme control. Ideally, I would have all the slides. I would define what the patch size is or the tile size is. I would define what the stride is and everything, so I can do all the analysis myself. Obviously, we are loading some public repository here, so we have to play by what data we have access to. Okay, now let's train our classifier. So we have 5 reference slides, let's say. How many slides did I put? Number of reference slides. We have 5 reference slides. And let's go ahead and run this first. And the 5 slides, we are extracting all the tiles from the 5 slides. Well, they all have tiles anyway. And let's see, one of these actually has, like T006 has 4,927 tiles, and the other one has 8,200 tiles. So you can see the range right there. Okay. And you may ask, why am I only taking 5 slides? Because KMeans on embeddings is surprisingly effective at separating tissue types with just a few well-chosen examples. So 5 slides give us enough diversity to capture the range of tissue appearances in the cohort without spending tens of minutes of time loading this entire data set. Yeah. Now, let's cluster the reference tiles. We have our reference tiles, right? And let's define our K equals to 5 as part of our KMeans. And let's actually cluster them using our KMeans from applied on all the 33,780 embeddings. Yeah, and that's not bad. The 5 clusters come roughly balanced, right? Between 5,000 right there and 9,000 tiles each. These are different clusters, by the way. In cluster 0, you have 7,000 tiles. Cluster 1, 7,000 tiles. And I'm looking at 5 clusters right there. Okay. And now let's actually do UMAP of reference embeddings colored by cluster. This one actually takes a little while, so let me go ahead and run that. So the UMAP supposedly should show us the 5 clusters. We saw that as part of the presentation, the 5 clusters projected onto 2 dimensions. Okay, and we already saw that these clusters actually are very nicely, tightly clustered. The red cluster right here and this one, the navy one upper left right there. And these are compact, isolated islands in the UMAP space. So there is some overlapping right there, diffuse overlapping, but in general, these are kind of tightly clustered. Okay, so now the question is, which cluster belongs to what? This is exactly where I would pick, like, I don't know, five tiles from this region, 5 tiles from this region, 5 from here, from here, and then I'm like, oh, then visually we can look at them and we can say, hey, these are background, and these are stroma, and so on. But we don't have that luxury. So that's exactly why we need to do some sort of a proxy. As you can tell, I added this slide later on without any notes. I added notes as part of the code right here. But this cell is, we don't have the tile images. So, this cell is like a proxy. Okay, so we are going to use clinical correlation as a proxy. Again, I explained the logic. The logic is tumor tiles should correlate positively with clinical severity, right? Higher stage. The tiles with tumor, they should correlate with higher cancer stage and higher mortality. Background tiles should show almost zero clinical correlation because glass, the background, carries no biological signal. And the stroma tiles should correlate negatively or neutrally with the cancer stage. So we're using that. So we predict the cluster assignment for every tile in all 420. In fact, this one is slow. Let's run this. We predict the cluster assignment for all of our tiles in 427 slides, right? And we compute the fraction of tiles in each cluster per slide. And then we correlate those fractions with the stage and 5-year mortality and MMR loss. Okay, and let's wait for the results so I can continue this explanation. And this is the only workaround I kind of managed to find. So let me scroll down and wait for this to reach 100%, and then I'll continue this. Okay, so looking at the results, what can I see? Cluster 3 right there has, it shows the strongest positive correlation of all, right? So with the stage, 0.124, and also the strongest positive correlation with mortality. So I am almost positive that this cluster 3 is the tumor cluster. Now, the cluster 0 shows almost zero correlation with stage, with 5. Yeah, cluster 0 seems to be the background here, because it's almost zero is what we expect from the background tiles, right? Which carry no clinical information. So this is our background cluster. Now look at cluster 1 and cluster 2 and cluster 4, right? So these are the ones. They show negative or near zero correlation with the stage and also the mortality, actually. Yeah. And in colorectal cancer, stroma-rich slides tend to have a lower stage, they tend to correlate or tends, towards lower stage tumors. And this pattern is consistent with stroma between all these. So we assign all these 3 to the stroma class. Again, this is my, in a way, educated guess. The right way to positively tell which cluster belongs to what is by looking at these images, which we don't have access to, unfortunately. Yeah, I would say cluster 2 is ambiguous right there of all, but stroma is the most plausible biologically, assignment, given that colorectal cancer tissue is typically 50 to 60% stromal. Yeah, that's why I'm comfortably assigning, let's say, with keeping that in mind, that this is the one that's a bit uncertain. I'm uncertain of compared to the others, like compared to cluster 1 and cluster 4. So that's it. So the resulting fractions. Let's see, what do we have? 26.8% tumor, 50% stroma, and the remaining about 20% is the background. And again, that's the biologically plausible for colorectal cancer. So I'm gonna leave them the assignments as is. So based on that cell, I'm going to assign these classes down here. Yeah, cluster 3. Where is my cluster 3? Right there, with a value of 0, which is our tumor right there. Clusters 1, 2, and 4 are stroma, and cluster 0 is the background. And now with these labels, I'm going to classify a logistic regression model on all the 33,000, how many ever tiles that we actually have. And let's look at the results. Yeah, 33,780. And tumor, again, about 9,000, 17,000 is stroma, background is 7,000, and the tissue classifier accuracy is 100%. That's, I mean, I'm not let's say, surprised with the accuracy here. I know the accuracy is going to be good. It's just that which one belongs to what is where we actually need to be careful. And I hope I assigned them correctly to these clusters. Okay. Okay. So now let's go ahead and compute the spatial features for all the slides. Yeah, this is the main computation. So let's go ahead and run this. So for each of the 427 slides, whole slides that we have, we load the embeddings, we scale them. Yeah, we need to scale them. And we predict the tissue class for every tile. Now that we trained the logistic regression with 100% accuracy, we can use this trained model, apply that to every tile from each and every one of our 427 whole slide images, and we kind of label them. Okay, that's what's going on here. And as you can imagine, this process will be slow, but not as slow. I mean, this is not bad. It's getting there. And yeah, let's wait for this to be done. And again, I'll continue once this thing is ready for our discussion. And by the way, one of the reasons why this is a little slow is, if you remember, spatial entropy is we're calculating spatial entropy right there, right? It bins the tumor tile coordinates into, our setting is 10 right there, into 10 by 10 histogram, and computes Shannon entropy. Okay, and that's a slow process, relatively slow process. And also the stromal adjacency is this is something we are looking at. It builds a KD tree right there. It builds a KD tree from tumor tile coordinates, and it queries distances from stroma tile coordinates. Now, with a radius of 512 pixels applied to coordinates that have 2013 pixel stride, right? I mean, in our case, from our coordinates, we saw that the stride is 2013 about 2013 pixels. Now, if you have a radius of 512, obviously it's going to find 0 neighbors for virtually every stroma tile. Yeah, so that's one thing that's expected. Okay, with that information, let's go down and look at the summary statistics right there. As expected, the stromal adjacency is 0 for everything. Yeah, there is nothing, the mean, standard deviation, everything. It's 0 for the reasons I just mentioned. Now, mean tumor fraction is 0.458, meaning mean stromal fraction is 0.542. Median TSR 50th percentile, 0.776 right here. Yeah, so that's fine. So we have all the features. And now let's go ahead and merge this information with the clinical labels. Okay, for this, now I'm going to add the clinical labels, and that's it. So yeah, everything looks okay. I thought I saw something. So 264 again survived, 161 died. That's the information. Now let's go ahead and visualize the tissue map. So we select 2 slides with the lowest TSR and 2 with the highest from the merged data frame. And for each slide, we load embeddings. We classify all the tiles. and we reconstruct the spatial map using coordinate rank ordering. Okay, and let's go ahead and do that. And the rank ordering fix is worth understanding. The raw coordinates at 40x, the raw coordinates at 40x, right? They have a stride of about 2000 pixels, 2013. And if we used raw coordinates as grid indices, a slide with 5,000 tiles would need a grid, thousands of pixels that's actually wide, where each tile occupies like one invisible pixel. I don't know if that makes sense to you. Anyway, go ahead and read this, the code, to understand exactly what we are doing. Basically, it ranks each unique. Let me skip that. Sorry, I tend to sometimes over-explain things. Let's go down and look at actually what the biology is and what the outcome is. So looking at these 4 maps, right? I mean, we picked 2 here, and we picked 2 with the lowest TSR, 2 with the highest TSR. Okay. Now, obviously, the left one, 539, case number 423. They have TSR values of 0.17. This one has 0.18. They're predominantly greenish or teal color here with a red border. You see, there are some red tiles right there, red border right here at the tissue margins. And this other side, cases 557 and 439 here, they have TSR values of 11.11 and 26.79. And they're predominantly red, obviously, with a thin occasional stromal rim right there. Okay, and this one 423 has large gray region right there with high background fraction, and this is almost like 40% background fraction right there. Yeah, this probably corresponds to physical tissue damage or gaps in the biopsy section. So the classifier correctly identifies this as background rather than misclassifying it as tumor or stroma. This could be stroma. Again, this is exactly where I would pull up a couple of images from this region and try to understand exactly what's going on. My honest guess is this is some tissue damage that probably looks like background or something. Okay. Now, let's actually do TME feature distribution by survival output, and we are going to just do some box plots here. And each TME feature, let's go ahead and look at the box plots. And the teal is survived, and this is the less than 5-year died. Yeah, who died within 5 years. So tumor fraction, the first one is showing us the tumor fraction. This one is the stroma fraction. And as you can see, first of all, they all show up with a p-value of 0.001, 001, 001, 3 stars right there. That means they're all statistically significant, right? So patients who died within 5 years have higher tumor fraction and lower stroma fraction. Yeah, this is exactly the published finding for colorectal cancer. Okay, and I think that's what I can. I mean, here these are not significant. You see, p equals to 0.1, p equals to 1.0. So no point in discussing about the spatial entropy, and of course the adjacency. We have nothing anyway. We have values of 0, so not much right there. Yeah, I actually find this spatial entropy part a bit interesting. It suggests that the spatial arrangement of tumor, it's not just the amount of tumor, right? It's a spatial arrangement of tumor carries some additional signal. Yeah. So yeah, that's it. And the Kaplan-Meier is skipped. This survival analysis requires a follow-up duration for every patient, including those who survived. And but we don't have that information. It only records days till death for the patients who died within that 5-year period of time. Okay, so I'm commenting out that part. And let's do the correlation heat map. So it computes the pairwise Pearson correlations between the 4 informative spatial features that we have, right? And the 3 clinical variables. So we're going to look at all these features: tumor fraction, stroma fraction, TSR, and spatial entropy. The other one, we dropped it. We don't have anything for the adjacency. And how do they correlate with stage, MMR loss, and the mortality right there. Okay. So the tumor fraction correlates positively with the stage, 0.16, with MMR loss, 0.13 right here, and with the diet. So we know this. We kind of earlier used this fact to actually positively tell that, hey, this is the tumor region. Yeah. We labeled it as such. Same with stroma. It's kind of exact mirror image of this. Actually, this is literally a mirror image. 0.16 minus 0.16, 0.13 minus 0.13. So this is a mirror image. This is pointing in the opposite direction. Yeah. And I think that's the end of it. This wraps our video 5B, and I hope you found this to be informative. We had some limitations there. I wish we had some tissue images to make this even more exciting, but we work with what data we actually have. So yeah, let's go ahead and meet in the next tutorial. And until then, again, please do not forget to hit the subscribe button. Thank you. Thank you very much.